All right. So before today, we talked about central dogma elaborations. We had gone through in the previous unit how we can get from all the way from DNA to protein. Now we need to talk about the processes by which that protein is shuttled around the cell, how that protein might be secreted out of the cell, and how sometimes that protein can be endocytosed by another neighboring cell. We're going to be talking about all of this in the context of a disease called Noonan syndrome. Noonan syndrome is a congenital disorder, which means it happens at birth. Some of the symptoms that emerge with Noonan syndrome is macrocephaly, which means an enlarged head. We briefly touched on Edwards syndrome, trisomy 18, which was displayed microcephaly. This is an instance in which a disease presents with macrocephaly. There's also hypertelorism, which is the eyes being slightly just further apart. And there's also a bit of a short webbed neck. In addition to that, there's also some cardiac defects that emerge with some of these patients. And so that can oftentimes, if left untreated, to a shorter lifespan. It is true that when these cardiac issues are treated therapeutically, that some of the patients can make it and have a much longer lifespan. The incidence rate for Noonan syndrome is about 1 in every 1,500 live births. That's on the same magnitude as cystic fibrosis, and so it's by no means its incidence is on par with some more well-known diseases. Its etiology has been identified and pinned down to defects in a specific signaling cascade, the RAS-MAPK signaling cascade. We're going to take a moment to try and walk through this because this is actually a pretty important thing for us for you all based on your projects, but also for you all to know as you move into, for a lot of you, into Ideas Lab next fall, where you'll actually be learning how to interrogate the MAPK signaling cascade, which I'm pretty sure is still one of the modules. So what's important to note is that at the top of this signaling cascade, so when we talk about signaling cascades, it's this, you can think of these string of events that, this cascade of events that the activation of one protein leads to the activation of another, leads to the activation of another, which then leads to some gene expression where a transcription factor is activated and then promotes transcription of some family of genes. So here in the RAS-MAPK signaling cascade, usually when you see the first and the last name of a signaling cascade, it's usually kind of like the first protein and the last protein of interest here. So RAS is a monomeric G protein. We talked about heterotrimeric G proteins. That is, by and large, these alpha, beta, and gamma subunits. There are some cases where G proteins exist as a monomer. And so RAS is one example of a monomeric G protein. Once activated, it'll activate another protein that's a kinase called RAF. If you guys remember, kinase, does anyone want to share with us remembering what kinases do? Again, this is an example of us reaching back and using info we've already gone over. What do kinases do? Phosphorylate, or whispering of phosphorylation. Thank you. Kinases phosphorylate proteins. And there's 3 specific amino acids that phosphorylation occurs on: tyrosine, serine, and threonine. We typically break up the species of these kinases by what they phosphorylate. So there's a whole family of kinases called tyrosine kinases. Dr. Kristen Nagley's lab studies tyrosine kinases exclusively. And so that's one family of kinases, those that specifically phosphorylate tyrosine on proteins. There's also another class of kinases. There are serine and threonine kinases. RAF is a serine threonine kinase. And so what's important to note is that we also used to call RAF, there's a larger family name that RAF exists under. It's called MAP kinase. It is, let me say this, right like this. It's a kinase that phosphorylates MAP kinase. And that's what MET is. MET in the next layer of the cascade is a MAP kinase. So what's important to note is that, again, it's a kinase, so its job is to phosphorylate. We're seeing a string of kinases acting here. It is a dual specificity kinase. What that means is that it can phosphorylate tyrosines and serine and threonines. It can act as both of those species, basically. And so, so MEK is also a MAP kinase, which means it's a kinase for MAP kinase. So basically, if you start, we have activator RAS. Let me go back to the computer so I'm heard properly. Forgive me. At the top, we have RAS that activates RAF, which is a MAP kinase, which then activates MEK, which is a MAP kinase. Sorry. And then which activates MAP kinase. All of these are families of proteins. So like under MAP kinase, which you guys may have come across in some of your projects. I think I've seen that in some of your captions or in your 3-minute theses. MAP kinase is a family of genes. So, or a family of proteins, forgive me. So some key examples that you guys might have heard of, P38, ERK, JUNK, those are all MAP kinases. So MAP kinase's job in this signaling cascade is to then phosphorylate some transcription factor. And so once that transcription factor is phosphorylated, now it's activated and can go and move from the cytoplasm into the nucleus and bind to the promoter of some set of genes that are associated with some processes that are tied to this signaling cascade. As I mentioned a moment ago, we talked about how G proteins are largely heterotrimeric. An exception to this rule are a class of monomeric G proteins. In this case, RAS is a monomeric G protein. But what still holds true for both monomeric and heterotrimeric is that they're both constitutively or always localized to the plasma membrane. We had shown in the G protein lecture, and you can go back to it, where there's an animation that shows the addition of a GTP molecule activates it in the alpha subunit, or in this case, it'd be RAS for the monomeric form, slides across the plasma membrane. When we go back to the fluid mosaic model back in Unit 1, we talked about how proteins can move around in the plasma membrane. They can kind of move and diffuse laterally. That's what these G proteins are able to do on the inner leaflet of our plasma membrane. Are there any questions here before we move on? All right. So we're going to take some time to try and to look at the trafficking of these proteins after they've been translated within the ribosome. And what's important to note is that we're going to talk about how some of these ribosomes, if you look here, are studded on the rough endoplasmic reticulum. That's going to be this organelle that's just outside the nucleus. That's going to be a key location for the synthesis of a lot of these proteins. Some of them are going to be in ribosomes. We're going to have some ribosomes that are free floating within the cytoplasm here. But I just want to take a moment to try and situate you all with where we've been spatially and where we're going. So we've been in these ribosomes. We're going to have proteins that are going to be synthesized in the rough ER, some of them in the cytoplasm on free ribosomes. And then we're going to look at how they can be shuttled out in some of these vesicles. And for some of these vesicles, what's key to note is that there are different directions of movement. So to take a moment, one thing we've talked about before, at least referenced previously, is this process of endocytosis. Endo meaning in. It's the process by which things that are outside the cell are brought in. And the process by which things that are inside the cell that are being released is exocytosis. You can think of exo as exiting. It's leaving the cell to the extracellular space. There are two directions of movement typically that revolve around transport. So when we have vesicles that come from our rough ER that move to the Golgi, which is another organelle here that we'll also talk about in a little bit. They'll go from the rough ER to the Golgi and then into these vesicles that move out away from the cell. That's called anterograde transport. All of the anterograde, you can think of it as moving from the middle of the cell towards the periphery. It's all situated relative to being within the cell. And so the opposite direction would be retrograde transport, where we're moving from the periphery, from around the plasma membrane towards the center of the cell. That is retrograde transport. Both retrograde and anterograde transport are relative to being within the cell. But when we talk about things that are moving inside the cell, moving in or out of the cell, that's when we talk about endocytosis or exocytosis. It's good to think about what you're talking relative to when you think about anterograde transport and retrograde transport versus endocytosis and exocytosis. These vesicles that help mediate some of this transport this is a scanning electron micrograph of one of those vesicles. And there's this coating within the pit of this vesicle that uses this group of protein, this protein they're called clathrin. It coats the entire inside of these vesicles, and it's pretty important. And the reason why is because it forms this structure, this ancient structure. It's called triskelions. It's this like 3-branch structure that all web together to form in this case, it forms a hexagonal, not prism, hexagonal shape when all the clathrin proteins come together with both their heavy chains and light chains. The heavy chains and light chains are indicated here in black and red. What ends up happening is that when something is being brought into the cell, so being brought in is endocytosis, there's actually going to be the budding off, the recruitment of these clathrin proteins to that site where something's being endocytosed. So one example is we talked about how there's receptor-mediated endocytosis for B cells when they're going to be bringing some antigen in to display because they're an antigen-presenting cell. So what ends up happening is that when a ligand binds its receptor, there's oftentimes going to be another protein that's called an adapter protein. Adapter proteins are just an entire class of proteins where they bring two things together that aren't necessarily interacting with each other, but just bringing them close together. An instance in which a protein brings things together and they actually interact is called a scaffolding protein. We're going to talk about a scaffolding protein later today, but I suspect a lot of you may have read or come across adapter protein and scaffolding protein, and it can be hard to distinguish what are those, how are they different. Here, an adapter protein just brings things together that might not necessarily be interacting with each other. So when the receptor binds to its ligand, this adapter protein is recruited in that'll then bring in clathrin. And so we'll have a lot of clathrin that comes together to form the early shape of this vesicle. And what ends up happening during this endocytosis process is starting at at number 1 here, we have the ligand binding to the receptor. When ligands bind to their receptors, oftentimes these receptors will aggregate together on the surface. Again, proteins in our plasma membrane can diffuse laterally. So these receptors cluster together. And as they do so, each of these receptors have an adapter protein that's bringing in clathrin. So we have the coating of clathrin underneath these receptors, and you're going to reach a point where they all bud together. What needs to happen, what ends up happening is that it forms this invagination of the receptors as they're being brought in by the formation of this clathrin coated pit here. But much like an apple on a tree, you can have something there, but you need to twist and pull it off. That's what needs to happen with this vesicle. So there's a protein here called dynamin that's boxed in here. It's a GTPase. So when when it hydrolyzes a GTP, it provides that pulling, that twisting and pulling, similar to pulling an apple off a tree here, that pulls the vesicle away from the membrane. And we have, at step 4 here, vesicle formation. So we have the clathrin coated on the outside. We have all these receptors with their ligands. What ends up happening is we lose this clathrin coating. It gets recycled for other receptors that are binding to ligands to endocytose things. So now we just have the membrane with these receptors and the ligands. What ends up happening now is that there's going to be a drop in pH that'll promote the dissociation of the receptor from the ligand. And the receptors will then be separated and be recycled to the cell membrane. And now we just have the We just now have a vesicle with just the ligand that bound to the receptor there. What happens next is going to depend on what kind of cell we're in. For example, if we're in, if this is an epithelial cell, those are the cells that provide the layering across a lot of mucous membranes in our body. They have a top and bottom side, an apical and a basal side. And sometimes they'll take things in from the apical side and release it on the basal side. That's called transocytosis. That's what 7C here is showing, where basically it's releasing the contents of that vesicle out to the other side. In other cases, it might be, you might have the formation of a lysosome that's just going to degrade what the contents are. So it kind of depends on what the cell is and what that receptor was that bound to the ligand. That's going to determine the fate of what was endocytosed. In the case of a B cell, that's an antigen-presenting cell. It's going to process that the antigen that was endocytosed to break it into pieces and display on MHC class 1 or 2, because it's an antigen-presenting cell. I see people whispering too. I see you and I appreciate you. That's correct. It'd be just processing it and displaying on MHC class 2. I know it's a really beautiful day outside and you guys would probably be rather out there than in here, but we'll get to that point soon. So again, the trafficking of these endocytosed contents are really critical. And we're also going to talk about how these vesicles also facilitate the process of exocytosis, or the process by which, when a protein is newly synthesized within the ribosome, how is it transported throughout the cell, depending on what its fate is of where it's supposed to go. There are key ways in which our cells know where these proteins, newly synthesized, newly translated proteins, are meant to go. There is two key examples of this process of moving them around or shuttling them around the cell. One is co-translational import. What co-translational import is at co-translational, so that means during the process of translation, the peptide that's being synthesized is being imported into some of these key organelles. The locations that these newly synthesized peptides will go, where they go will determine whether it's co-translational import or not. For those that are going to end up in the extracellular space that are going to be secreted, for those that are going to end up on the cell membrane, or for those that might end up on a lysosome, those are the proteins that will undergo co-translational import. For those that are going to end up in other locations, like the nucleus, like the mitochondria, or just in the cytoplasm, that's going to be post-translational import. They're going to be undergoing that trafficking after translation is complete. What determines where they go is actually a signal that's embedded in the amino acid sequence, in the primary sequence of that peptide, and it's going to serve as that, so that it's an amino acid sequence or a motif. When you hear motif, that's going to be a specific sequence of amino acids that'll determine where does this newly synthesized protein go. So in the absence of any of those signals, that protein is just going to end up in the cytoplasm. And there's lots of proteins that are floating in our cytoplasm at any given moment. There are key ones. You know, for example, the actin cytoskeleton. Those are always going to be in the cytoplasm of our cells. That are incredibly important. And so in that case, those that are going to be destined to be in the cytoplasm lack any sequence at all. For the co-translational import, they're going to have a specific sequence that we call the signal sequence. The signal sequence is going to designate where that protein is going to go, whether it's going to be out to the cell membrane or outside the cell. And a specific targeting sequence can determine if they might go elsewhere, for example, like a lysosome. If it's going to be post-translational import, there's going to be one of two signals. There's either going to be a nuclear localization signal, that means it's going to be destined to go to the nucleus, or a mitochondrial targeting sequence, that it's going to be destined to go to the mitochondria. So for post-translational import, it's much more clear. It's either going to go to the nucleus if it has a nuclear localization signal, or the mitochondria if it has the mitochondria targeting sequence. If there's no sequence at all, it's going to go to the cytoplasm. For co-translational import, we're just calling it the signal sequence. There's going to be another sequence that we talk about during, not necessarily sequence, but for those that end up in the membrane, there's going to be a sequence of 20 hydrophobic amino acids. They're going to determine that's going to be embedded in the plasma membrane. In addition to that, proteins that are typically smaller than about 40 kilodaltons are going to freely diffuse in and out of the nucleus. And so there's going to be, that free diffusion is going to play a role in the nuclear localization that we'll revisit. So to take a moment to focus on co-translational import, what ends up happening is that we have the key signal sequence that is, again, it's an amino acid sequence in this polypeptide. It's going to be recognized by a protein that's called the signal recognition particle, or the SRP. So it's going to come in and recognize that signal sequence. And it's going to hit pause on translation for a minute. So we have a ribosome here that is just bound, that has just started translation. If that signal sequence is present and recognized, it's going to push pause on translation. That ribosome will now be brought over to the ER membrane, where there's a receptor for SRP, the SRP receptor, and there's going to be a ribosome receptor that's just going to recognize the full ribosome assembly. Once all of that docks, we're going to have the GTP that's there's going to be a GTP here that binds to the SRP receptor. When that happens, the peptide is passed from SRP into a pore here on the surface of the ER. Once that GTP is hydrolyzed, SRP leaves, and once it's released, now the polypeptide, we hit play on translation, and the polypeptide can grow into the lumen of the ER. Once it grows into the lumen, what I should also say is that the N terminus, the front end of the polypeptide, is going to be kind of looped and stuck in the pore, so that it kind of helps keep it there during translation. What ends up happening is that as we get to the end of translation, we talked about how there's the termination factor that cleaves the C terminus. Well, at the N terminus, there's a signal peptidase that'll cut at the N terminus here, and that'll release a fully formed peptide, polypeptide, into the lumen of the ER. This is what this is an illustration of what the ribosome on the ER actually looks like. Hopefully this works. It does not. Forgive me. And then this is a barrel and ribbon illustration of what the pore protein actually looks like as well. I want to stop here and leave space for a couple of questions about co-translational import. Yes. Mm-hmm. Is that separate from what, sorry? Is that separate from what happens during translation? So like all of the steps of translation are still occurring. Like everything we talk about translation is situated with just the transfer RNA and the ribosome here. All that's happening now is that this ribosome is docked at the ER, and this happens with a lot of proteins. So if we're talking about proteins that are, so for co-translational import, extracellular proteins, and cell membrane bound proteins, that's a lot. So this is a pretty normal part of where translation can happen, where the ribosome is physically located. Does that help clarify? It's unique to co-translational import, yes. Does that help clarify? Great. Okay. So another key example of co-translational import that's important for us to discuss is those that have a key stop transfer or topogenic sequence. In this case, it's one where there's a sequence of 20 hydrophobic amino acids that are going to have that are going to determine that this is going to be a protein that has a region that is embedded in the plasma membrane. If you go back and think on the early lectures in Unit 1 where we talked about a hydropathy plot, and there was a protein called tetraspanin that had 4 regions that went in and out of the cell membrane. Each of those regions that went into the cell membrane were 20 amino acids, at least 20 amino acids long. And so that is a stop transfer sequence, those 20 hydrophobic amino acids. What ends up happening is that as the stop transfer sequence is recognized, what's going to end up happening is that the ribosome that was on the ER is now going to undock from the ER and be free floating nearby in the cytoplasm. And what's also going to happen now is that the protein will now be synthesized into the cytoplasmic space as displayed here. With each 20 hydrophobic amino acid stop transfer sequence, that's going to be moved laterally out of the pore into the membrane to help build that transmembrane protein. So if we think back to a protein like tetraspanin that had 4 regions that were wedged within the membrane, as it's being synthesized, whenever that stop transfer sequence is recognized, it's going to pass it laterally out of the pore into the membrane and continue synthesizing this polypeptide until, as with co-translational import, the signal peptidase will now cleave at the N-terminus, releasing this protein. And in the ER membrane here, we'll have this protein will now be embedded in the ER membrane. There'll be a process now by which from the ER membrane, it's going to be trafficked and moved out to the cell membrane. So that was co-translational import. Another key thing to note is that once these proteins are in the ER, there's going to be some key post-translational modifications. It's a big term that just means after translation is done, there's going to be some modifications to this new protein. And there's going to be 5 key kinds of modifications that exist that I want you guys to note. There can be the formation of disulfide bonds. The ER is an oxidizing environment, and so the existence of these cysteines with the thiol groups on them will promote the formation of disulfide bonds. There's going to be the proper folding that happens in the ER. Again, biology exists in 3 dimensions, and so as it's being synthesized, the hydrophobic regions are going to be interacting with each other, promoting the folding of this peptide as it's translated. There might be specific sites where things need to be proteolytically cleaved. Going back to the extracellular matrix lecture, we had talked about how some ECM proteins like collagen type 4 and laminin are synthesized in a precursor form that need to be cleaved into their mature form. And so in some cases, examples like that those are examples where proteins need to be cleaved. It's also where multimeric protein assemblies are brought together. And finally, it's where the addition of sugar groups or glycosyl groups are added to these proteins. The process of adding these sugar groups is called glycosylation. What some of this glycosylation can look like, I'm going to take a moment here to say, I'm not asking you to memorize this entire tree here, but rather trying to help categorize 2 kinds of glycosylation that are going to be helpful, that help illustrate this. So whenever you see saccharides, that's usually like a sugar molecule. You might hear monosaccharide, which means 1, disaccharide, which means 2, oligosaccharide. Oligo, I think, is a Greek prefix that just means like several or a few. So what we're going to have here is the addition of oligosaccharides during glycosylation, where there's going to be a few sugar molecules that are added at different sites on a protein. Those sites are going to determine what kind of glycosylation it is. So, for example, if we're at a carboxyl group where we have a hydroxyl a hydroxyl group within that carboxyl group where there's free oxygen, on that oxygen, if there are oligosaccharides added, that's called O-linked glycosylation. There's going to be the addition of these oligosaccharides to the oxygen on that hydroxyl group, usually going to be usually on a serine or threonine. And these O-linked glycosylations are usually pretty short. We have in the left half of this tree of this figure here, we have O-linked glycosylation, where there's the hydroxyl group here that's on the carboxyl between these two amino acids, and it's there's going to be the addition of just a couple of saccharide groups here, whether that's on collagen or another glycoprotein like glycophorin. The other example of O-linked glycosylation is N-linked. So now at the other end, we have the amine group at the N terminus or towards the N terminus, where the nitrogen on the amine is where these sugar groups are going to be added. And so this is going to be much more highly branched and much more longer additions. And so whereas we had two examples here of just a few sugar groups being added, all of this on the right half is one glycosylation, or not one, the branching of many glycosylation events off of one single amino acid here. And so this is the degree of complexity that can come with, that occurs with N-linked glycosylation. The glycosylation events that occur, they can start in the ER, in the lumen of the ER. Some of these events are going to happen as well as in the Golgi. There's going to be different flavors of glycosylation events that occur in these different organelles. And it's going to happen and be mediated by a series of enzymes that add these sugar groups. Some examples might be, one example is a galactose transferase that I think we'll talk about in a little bit. But there's a key thing there is that these enzymes mediate all of this glycosylation. And each of those enzymes are going to be restricted to certain organelles. So like different flavors of sugar molecules are going to be added in different parts of the cell. And the entire, and this also happens for proteins on our cell membrane, because our cell membrane is going to have lots of glycoproteins on the surface. As we refer, just like with histone modifications, where we call the histone code, all of the histone modifications, when we look at a cell and look at all of the glycosylation on the surface of a cell, we usually refer to that as the glycocalyx. So some of you might be thinking, it's just sugar being added onto a protein. What does that actually matter? What is that functionally, what's the functional relevance of this? One example of why this could really matter is that, as you all have learned, this is Western blotting from our protein biochemistry lecture. We have two different monoclonal antibodies here that both stain for GDF11, that's for the same protein. So they stain for different epitopes on the same protein. What ends up, if you can appreciate here, the way these bands emerge on the Western blot look different between these two monoclonal antibodies. What ends up happening is if you inhibit the, and you typically with Western blotting, you want a nice clear, you want to have nice contrast and have a very clear black band, not some of the smearing you see here or over there. So the question became, could this be an issue with glycosylation that's blocking the epitope of this antibody? So using purified pro-GDF11, when you expose them in a normal condition and run them, you get a smeared band. When you use an inhibitor that blocks glycosylation, you get a very clear band. That's true with both this monoclonal antibody for GDF11 and this one. So the reason why this matters is that a lot of therapeutics use monoclonal antibodies. And even in a research setting, these glycosylation events block these epitopes. And it's possible that the efficacy of some monoclonal antibody drugs might be inhibited by glycosylation events that we might not be aware of. Another example that might be a little more relevant for the pre-med group in here is the glycosylation matters as it relates to our blood types. the way in which our blood types are determined are by expression of a certain gene that is a sugar modifying enzyme. It's a glycosyltransferase. So if you have a blood type, the kind of allele for this gene, allele means like the kind of the copy of this gene. The A allele for this gene expresses GalNAc transferase. It adds it adds an N-acetyl galactosamine at the end of this glycolipid that's on the surface of our cells. If you express the B allele of this gene, you have a galactose transferase that adds galactose. If you have O blood type, you do not express a functional enzyme. You have a frame shift mutation where there is no enzyme expressed, and you have the bare glycolipid here without either the galactose in front. So you can tell that there's the same structure here before this galactose here, or the N-acetyl galactosamine here. But the O type lacks the enzyme, and that's why typically O blood type is suitable for all patients. We went 2 minutes over. I apologize. I'm going to stop there. Thank you very much, and I hope you guys have a good weekend and enjoy the weather.